Good evening. Tonight we're going to talk about cell cycle regulation in the cell. The length of the cell cycle is variable. Cell cycle lifetime and the cell phases. Cell cycle length is highly variable. The frequency at which cells turn over can range from a lifetime spent in G0 to 2 to 5 days for epithelial cells to a few hours for embryonic development. Variation also exists in the time spent in each phase. Mammalian cells outside of the body can only spend about 24 hours for the entire cell cycle. This would give you 9 hours for G1. S phase would last about 10 hours. That would leave about 4.5 hours for G2 phase, and finally a half an hour for M phase. Regulation by external events. Extracellular events inhibit and initiate what's happening. Cell division is both initiated and inhibited by extracellular events at the onset of the replication process. One such event could be the death of nearby cells or the release of HGH, which is the human growth hormone. A lack of human growth hormone can inhibit cell division, which could lead to dwarfism. Or too much human growth hormone could lead to gigantism. Cell crowding can inhibit cell division also. A factor that could initiate cell division is the size of the cell. As it grows, it becomes more inefficient because it has decreasing surface to volume ratios. So the solution is for the cell to divide so that it can be more efficient again. You look we've got a cell, it's dividing, and then this is potentially a cancerous cell, and then it's dividing into malignant cells, and they're dividing abnormally. So, regulation of internal checkpoints. We're going to talk about daughter cells. We're going to talk about chromosomes. We're going to talk about duplication. We're going to talk about control mechanisms. We're going to talk about checkpoints that occur. We have 3 checkpoints. They're notated here on the slide. We notated here some points that happen during the cell growth checkpoint, during the DNA synthesis checkpoint, during the mitosis checkpoint. We're going to talk about each of these one by one. So each of the daughter cells has to be an exact duplicate of the parent cell. Any mistake made during duplication or distribution of the chromosome will be passed forward from then on. There are internal control mechanisms to prevent compromised cells from continuing to divide. The control mechanisms operate at 3 main cell cycle checkpoints. In a eukaryotic cell, a checkpoint is a point in the progression of the cycle to the next stage where the cycle could be halted until all conditions are met to continue. The checkpoints are located near the end of G1, at the G2M transition, and in metaphase. And you can see them here. Near the end of G1, this is the one in metaphase, and this is the one in the transition here. G1 is irreversible. It does a DNA check for damage, and it has external influences. We do a little bit of diagramming here. Here's our G1. It says that it looks at cell size, the nutrients, DNA damage, and growth factors. So at this checkpoint, the cell irreversibly commits to cell division. The external influences previously discussed, such as growth factors, play a huge role in moving the cell past G1. At this checkpoint, there is no DNA synthesis, but there is a genomic check for DNA damage. RNA and protein synthesis still go on. If the cell does not meet all requirements, it will not move on to the S phase. At this point, the cell can halt the cycle and attempt to correct the issues, or advance to the G0 phase and wait for further signals when the conditions improve. So if it can't fix it, it's going to go into G0 and it's going to wait. And it could sit there for a lifetime if it never is able to fix it. Or what can happen is it could receive a signal to just destroy itself. Otherwise, the DNA damage could be fixed, or if there's nothing wrong with it, it's going to move on to the S phase. G2, meiotic phase. Cell size, DNA damage. And that one's over here. It's a checkpoint, cell size, DNA replication. If conditions at this checkpoint are not met, the cell cannot enter the mitotic phase. As with the checkpoint G1, cell size and protein reserves have to be adequate. For this reason, RNA and protein synthesis continue, and the cell just about doubles in size. The most important check at G2 is ensuring that all the chromosomes have been replicated and that the DNA has not been damaged in the process of replication. If DNA damage is found, the cell is halted. The cell at this point attempts to complete DNA replication or repair the damage. Again, if it cannot repair the damage, it'll go into G0 and wait for further instructions, or if the damage is bad enough, it'll receive the signal to destroy the cell so that there is no risk of duplicating the damaged cell. The last of the checkpoints is the M phase, the M checkpoint. This checkpoint occurs near the end of metaphase. It is the spindle. It is also known as the spindle checkpoint. It's this one right here. It looks for chromosome attachment, and this is because this is where it is verified that all the sister chromatid are attached to the microtubules properly. This checkpoint is crucial because the separation of the sister chromatid in anaphase is irreversible. This will halt the cycle until all sister chromatid are properly attached to at least 2 spindle fibers. Because if they were to separate and the sister chromatid were not attached properly, the cells would not get the appropriate DNA that they needed to be effective. Let's talk about regulator molecules. There's positive regulation, negative regulation, and they work with regulatory proteins. There's enzymes that get involved. a whole host of things, and there's complex protein complexes that get involved. We're going to talk about them all. Okay, so there are two groups of intracellular molecules that help regulate the cell cycle in addition to the checkpoints. The molecules are responsible for either promoting progress, which is positive regulation, or halting the cycle, which is negative regulation. Regulatory molecules can act alone, or they can influence the production or activity of regulatory proteins. So some of your kinases are going to be your enzymes that regulate other proteins. They got to be phosphorylated. Okay, adding a phosphate can activate or inactivate them. Cyclins are proteins that control the progression through the cell cycle. Cyclin dependent kinases, we'll talk about those too. So let's talk about positive regulation of the cell cycle. We have cyclins, we have cyclin-dependent kinases, cyclin proteins, internal and external signals. So in our image here, we're showing cyclin-dependent kinases, and we're looking at our target proteins. We're seeing our CDK, we're seeing, which is our cyclin-dependent kinase. We're seeing our cyclin here. We're seeing a complex where they're binding together. Here's the phosphate that's phosphorylating them. And here's our target protein. So this involves two groups of proteins, cyclins and cyclin-dependent kinases, which we're going to refer to as CDKs. These are the regulators responsible for the progress of the cell through the checkpoints. Levels of the four cyclin proteins, A, B, D, and E, tend to fluctuate throughout the cell in a pattern. Increases in cyclin concentrations are affected by internal and external signals. When the cell moves to the next phase, the active cyclins are degraded by enzymes in the cytoplasm. Cyclins can only regulate the cell cycle when they are tightly bound to CDKs. Each cyclin CDK complex has to be phosphorylated in a specific location to be active. CDKs are enzymes, kinases that turn into phosphorylate and other proteins. The phosphorylated cyclin CDK complex is active in advancing the cell to the next phase. CDK protein levels are stable throughout the cell cycle, but cyclin concentrations fluctuate, so they determine when the cyclin CDK complex is formed. Just like in a chemical equation, there's a limiting reactant. The cyclin would be the limiting reactant. It's going to be the one where its levels fluctuate, so it's going to be the one that's going to determine when the cyclin CDK complexes form. So cyclin levels are based on the timing of the cell cycle, not on specific events. So groups of proteins called cyclins, CDKs, we talked about that. They're responsible for moving the cell through the checkpoints. Cyclins control the cell cycle only when they are tightly bound to your CDKs. We talked about that. Like all kinases, CDKs are enzymes that phosphorylate other proteins. We talked about that. And the proteins phosphorylated by CDKs are involved in letting the cell process to the next phase. So negative regulation of the cell cycle. The best understood negative regulatory molecules are retinoblastoma proteins, P53 and P21. Retinoblastoma proteins are common in many cells and are a group of tumor suppressing proteins. Much of what we know about cell cycle regulation has been acquired from research done on cells that have lost regulatory control. In cells that have been found to be replicating uncontrollably, all three of the proteins have been damaged. So, your retinoblastoma proteins, a group of tumor-suppressing proteins common in many cells. They act primarily at the G1 checkpoint. P53 is a multifunctional protein that has a major impact on the commitment of a cell to division. It acts when there's damaged DNA in the cells, mainly at the checkpoint for G1. If damaged DNA is detected, P53 is the one that halts it. We'll talk about that more in the next couple slides. And we'll talk about this last point on the next couple slides also. So let's talk about negative regulation of the cell cycle from the point of view of P53 and P21. We're going to talk about how they mainly are at the G1 checkpoint, how they deal with damaged DNA, and how they work with apoptosis. The RB, P21, and P53 proteins act mainly at the G1 checkpoint. P53 is a protein that halts the cell cycle at G1 if damaged DNA is detected. P53 also calls in enzymes to repair that DNA. If for some reason the DNA cannot be repaired, P53 can trigger apoptosis or cell suicide in that cell to prevent duplication. Destroy the cell so that there is no way that it can possibly be duplicated. As levels of P53 increase, it'll trigger the production of P21. P21 is the enforcer for the halt dictated by P53. This is done by P21 binding to and inhibiting cyclin CDK complex activity. As both P53 and P21 levels increase, the chances of the cell moving into S phase decrease dramatically because they're binding up the cyclin and the CDK, which pushes the progression of the cells. So if they bind those up, the cells aren't going to be able to move forward through the cell cycle. So retinoblastoma proteins use its regulatory influence on other positive regulator proteins based on cell size. In an active, dephosphorylated state, retinoblastoma will bind to transcription factors. There are a variety of proteins. The transcription factor it most commonly bonds to is E2F. Transcription factors turn on or trigger specific genes, allowing proteins encoded by that gene to be produced. RBE2F complexes block the necessary proteins for the G1S transition. As cell size increases, retinoblastoma is phosphorylated to a point of inactivity. So as it gets phosphorylated, it gets to a point where retinoblastoma cannot be active any longer. At that point, E2F is released, and it is free now to turn on the genes to produce the transition proteins needed to get the proteins produced for the G1 to G1S transition.